Look closely at the texture of a ripe mango's skin. The vibrant colors and high sugar content we associate with the modern commercial fruit are actually agronomic anomalies extracted from a plant with a highly volatile genetic history. Mangifera indica is a highly complex alloploiploid, a species possessing multiple sets of chromosomes derived from different ancestral lineages. Morphological and pollen studies trace the precise center of this species formation to the continental regions of Burma, Thailand, Indochina, and the Malay Peninsula, extending through the Sunda Islands. Over thousands of years, the specific environmental pressures of this Southeast Asian geography forced immense natural genetic variability. Constant cross-pollination in the wild created a massive spectrum of localized traits. This table shows the chemical composition of ripe mango varieties, highlighting this genetic drift. In the rightmost column, carotenoids, responsible for vitamin A, swing wildly from 800 micrograms in Neelam up to 17,000 in Alfonso. For a wild plant, this extreme genetic diversity ensures survival across diverse climates. For an agriculturist attempting to harvest a consistent, uniform crop, it presents a massive liability. India alone possesses roughly 1,000 distinct mango varieties, resulting from natural crossing and gene mutation. Yet the commercial mainstays cannot be reliably grown simply by planting their seeds. The mango is highly heterogeneous. This means its seeds inherit an unpredictable, highly mixed combination of genes from cross-pollination between two distinct parent plants. If you plant the monoembryonic seed, a seed containing only a single embryo derived from fertilization, of a premium Dashahari fruit, the resulting tree will not produce Dashahari mangoes. It will yield a genetically unpredictable offspring, almost certainly bearing inferior fruit. To achieve commercial viability, agronomists must bypass natural seed reproduction entirely. The modern orchard is built through strict environmental manipulation and biological engineering. Extracting a predictable yield from an inherently unpredictable plant requires controlling every variable, starting from the dirt up. The first step of orchard engineering is selecting commercial varieties that map perfectly to the specific climatic limits of a geographic region. Northern mainstays like Dashahari and Langra are mid-season yielders, requiring specific subtropical cues, while southern and western varieties like Alfonso and Neelam possess distinct physical tolerances suited for different temperature gradients. The commercial varieties of one region often fail entirely when moved to another. Regardless of the variety, the physical soil parameters are absolute. The tree requires a deep rooting zone, providing 2 to 2.5 meters of clearance in highly well-drained earth. The chemical limits of the soil are equally strict. The substrate must be slightly acidic. Cultivation becomes unprofitable in soils exceeding a pH of 7.5, which eliminates highly alkaline regions and dense black cotton soils from commercial viability. Even with perfect subterranean conditions, the tree faces severe above-ground climate threats, peaking during its highly vulnerable flowering phase. Orchards located in microclimates that experience frequent rains or humidity exceeding 80%, particularly when accompanied by low temperatures, face catastrophic crop failures. These specific atmospheric conditions trigger lethal fungal pathogens. Powdery mildew and anthracnose will physically coat and necrotize the panicles, the highly branched flower clusters that bear fruit. If the foundational soil chemistry and the local microclimate are not perfectly aligned, the crop succumbs to disease long before a single fruit sets. Because seeds cannot transmit superior genetics reliably, commercial planting relies strictly on cloning via asexual propagation. Traditional methods of propagation, such as inarching, are highly inefficient. They require the cumbersome fabrication of raised wooden platforms around a mature mother tree just to place potted rootstock seedlings within reach for grafting. Modern agronomists use veneer grafting. This technique joins a detached scion from a superior variety directly to a rootstock. The vertical flap of the rootstock bark is retained and tied firmly over the scion's sloping cut to fuse their vascular tissues. For rapid plant multiplication, nurseries use an accelerated technique called epicotyle, or stone grafting. This aggressive intervention involves beheading an 8 to 15-day-old germinated seedling. A wedge-shaped scion is then driven directly into a fresh vertical split in the severed rootstock. To prevent the fresh vascular tissues from drying out or sustaining sun damage,
The grafts are sealed in polyethylene tape and housed in semi-shade. In essence, grafting effectively manufactures a biological cyborg. It leverages the robust root system of an unrefined seed to strictly deliver the engineered genetic payload of a commercial clone. Once it is fully established in the orchard, the newly grafted sapling will rapidly push out fresh vegetative leafy flushes. To maximize this growth, the agronomic formula for nutrient loading shifts dynamically. Exact chemical inputs must be precisely calibrated against the specific age and life phase of the tree.
This graph models application rates for a young orchard. For rapid canopy expansion during the non-bearing phase, a tree requires 73 grams of nitrogen, 18 grams of phosphorus, and 68 grams of potassium for every year of age. However, excessive application of nitrogen without a carefully balanced supply of potassium creates an elemental bottleneck, inducing severe potassium deficiency and crippling the plant's photosynthetic capacity. As the tree matures into its bearing age, the nutrient strategy pivots. The primary objective is no longer to grow leaves, but to force the production of fruit. Achieving this requires a critical physical intervention. 2 to 3 months prior to the expected flowering period, irrigation is halted entirely. Withholding water terminates the vegetative flushing phase. This calculated environmental stress signals the tree to shift its energy reserves toward reproductive survival, promoting active flower bud differentiation. Commercial cultivation is a precise mechanical cycle of fueling rapid growth, abruptly followed by calculated starvation to trigger the tree's reproductive responses. Look closely at the structure of a blooming mango panicle. The tree produces thousands of these tiny flowers per branch. Yet, despite optimal soil chemistry, flawless grafting, and precise fertilization, the ultimate yield of the orchard is severely throttled by the tree's highly inefficient reproductive biology. The architecture of the inflorescence is overwhelmingly biased. A panicle generates 1,000 to 6,000 flowers, but the vast majority are strictly male. Only a tiny fraction are perfect, hermaphroditic flowers capable of producing fruit. In varieties like Romani, perfect flowers account for merely 0.74%. Furthermore, the heavy pollen of Mangifera indica is not carried by wind or gravity. It relies entirely on specific dipterous insects, like hoverflies, to physically transport pollen grains between flowers. The tree expends massive biological energy, generating thousands of blooms, while structurally restricting itself to a fraction of a percent of potential fruit. Even if a perfect flower is successfully pollinated, it faces a final, devastating biological hurdle, the phenomenon of self-incompatibility. If a perfect flower receives pollen from its own tree, rather than being cross-pollinated by an insect from a different tree, the fertilization process initiates a doomed sequence. The self-pollinated ovule successfully fertilizes, but it invariably aborts and drops from the tree within 4 weeks. None of these selfed fruitlets ever reach the half-grown stage. The physiological cause is systemic. The self-pollination triggers abnormalities in the proembryo and causes an overgrowth of nucellus cells, which actively block nutrient pathways to the developing seed. Conversely, cross-pollinated fruitlets possess higher RNA and DNA levels. They act as a strong physiological sink, an area of active growth that aggressively draws in and accumulates metabolites, pulling the required nutrients to survive. Because of these biological bottlenecks, the final retention of mature fruit per panicle is staggeringly low, dictated entirely by the stubborn genetic characteristics of the chosen variety. Mango cultivation is not a process of passive, traditional farming. It requires the rigorous, active management of a flawed alloploiploid organism, forcing an incredibly inefficient biological system to consistently yield a global commodity.